Welcome, folks, to Chapter 10 of Microbiology. We're going to continue our discussion of microbial control. So we had previously discussed microbial control in the environment. Now we're going to move to microbial control within the body, essentially post-infection. Essentially, we're really going to be discussing antibiotics today. So what we're going to do is take an overview of kind of the different mechanisms by which antibiotics can function, and then we're going to focus on specific examples of those mechanisms. And so specifically in Chapter 10, figure number 2 goes through the different types of mechanisms that we'll cover and many examples for each of those mechanisms. If you look at the first link in the description, you'll see highlighted just specifically the antibiotics that you'll be responsible for that we're kind of focused on for this discussion to simplify it a little bit, okay? So that's the first link. What I'm going to recommend for you to do is actually generate your own flowchart. Now, this can be as simple as an outline or as complex as a diagram that actually includes images as well, so that you can keep track of the different mechanisms that we talk about and some of the examples within them. Okay, and that's what we're going to actually submit for participation points, and that's the second link that's in the description. The third link that's in the description is a link to the Ninja Nerd series on antibiotics. It's a really nice and engaging discussion of different types of antibiotics. It is a little bit more detailed than what you'll need to know for this class, but it does give you a lot of good perspective and more information on some of these different antibiotics as well. So I recommend that as a resource. So feel free to go ahead and pause this video, review previous notes, take a peek at the outline for Chapter 10 and prepare yourself for some sort of flow chart, either an outline or a chart that you're actually drawing out, so you can keep track of the different mechanisms that we're going to be talking about today. And this is that figure 2 that talks about kind of those different mechanisms and gives you an idea of what we're going to be talking about today. So go ahead and pause the video, come back in, and we'll go ahead and start. Okay, so we've talked a little bit about antibiotics when we talked about the history of microbiology, right? And specifically penicillin. So Alexander Fleming was kind of looking at his plates and his plates that he was growing microorganisms on, and he noticed that there was this fungus, and around the fungus, there were no bacteria growing. We call this a zone of inhibition. And that makes a lot of sense because in the environment, we've discussed this, that in the environment, microorganisms are constantly interacting with one another. Some of those interactions are beneficial, some of them are, negative and competitive. And so it would make sense that in the environment, microorganisms would have the capacity to try to outcompete one another. And so they do produce chemicals that naturally outcompete other species of microorganisms. And then as human beings, we then take advantage of that production of chemicals and isolate those chemicals. So we can either use them in their natural form, just as they are produced by a fungus or a bacteria or another microorganism, or we can have semi-synthetic versions of those where we collect an antibiotic and modify it slightly so it's more effective. Or we can actually produce synthetic analogs, analog being a similar chemical that has similar kind of impact, but is completely synthetic and lab-made. And then those different antibiotics can be used to control microbial infections in us as human beings, right? And so a lot of these different antibiotics that we'll discuss will come from these natural sources or be modified off of natural sources. So in your book, you have this table that talks about some of the antibiotics that come from fungi and from bacteria, and we will talk about a number of these in our discussion today. Just a quick note on formatting. As you know, scientific names should always be italicized when they're in kind of a PC format. In this case, PowerPoint did change that formatting, so I wanted to make sure to note that as we're here. Okay, so let's start chatting about some of these antibiotics. First, a general discussion. When we're using an antibiotic, remember that antibiotic means against life, right? And so anything that impacts another living system, we have to be very thoughtful of how it's going to impact the host, because us as humans, we are living systems. We also know from our discussions in this class, homeworks, and other discussions that we'll have around this class, that our individual self as a host is also accompanied by a very large community of microorganisms. So thinking about how those chemicals are going to impact us as the host and also our community of microorganisms, which is our microbiota, is really essential in determining which of these antibiotics that we need to use. Sometimes the antibiotic is specific to a specific type of disease, and that's kind of what we have. So we'll talk a little bit more about how we select antibiotics at the end of our chapter today, but thinking about really being aware of the toxicity of antibiotics is important. And what you'll see is a lot of the antibiotics that we talk about today will target processes that are specific to prokaryotes, right? We will focus on bacterial pathogens in our discussion today. We'll talk a little bit about fungal pathogens and a little bit about antivirals as well. But we will focus on antibiotics that specify or specifically target bacteria, generally speaking, in this discussion. Anything that is an antifungal, for example, or a chemical that's for the control of a protozoa, for example, is likely to have more of an impact on the host as well, just because those are eukaryotes versus prokaryotes. So we'll look at antibiotics that are specifically targeted to prokaryotes in this discussion, but have that distinction in mind. Okay. So we're going to look at essentially we're going to organize this lecture by looking at different mechanisms of impact. So here you see a bacterium, and it has genetic information inside. Then it has the metabolic processes that are kind of implementing that genetic information around it in the cytoplasm. And then it has some sort of barrier between it and its environment, right? Cell membranes, cell walls, for example. And so antibiotics will work in different ways. Some of them will impact the genetic code, right? And it'll impact replication, transcription, translation, right? So here you have replication and transcription, and over here you have protein synthesis. Some of them will impact the metabolic activity that's occurring within the cell. So here we're going to specifically look at the folic acid pathway, but there are other pathways like protein synthesis and other different types of metabolites that are essential to the cell that can be targeted by antibiotics. You can also have antibiotics that target that boundary between the cell and its environment. If the cell is not able to maintain a boundary between itself and its environment, then it cannot maintain its metabolic functions, and therefore it can also not maintain homeostasis, and therefore it cannot maintain its living processes, and then that cell will die. Okay. Now, we will have this discussion about antivirals a little bit that looks specifically at attachment of viruses or release of genetic information of viruses from their capsules. Just a brief discussion on this as well. And so essentially, those are the different mechanisms that we're going to focus on. And then the link that you have below specifies, it's just the same image that you see here, specifies which antibiotics we'll use specifically as examples. And those are the ones that will get into kind of the meat of how they work. And those will be the ones that you'll want to make sure to review for this chapter. Okay, so let's start with the cell wall. What's the purpose of the cell wall? The cell wall is a barrier between the cell and its environment. It's also a protective layer around that cell. So anything that perturbs the cell wall is going to actually expose that cell to any toxins or the environment, or just disturb the integrity of the cell altogether. And this is what we'll see, is that initially these antibiotics will disturb the integrity of the cell wall, and then that cell wall will essentially lose its function, and then there's no way that that cell can maintain its integrity and will actually start to lose its own cell contents because that cell wall is no longer functional. Okay, and the two types of antibiotics that we'll talk about are penicillin, which is a beta-lactam, and also vancomycin, which is a lipoglycopeptide. Okay. And so let's look at the cell wall for a second. This is what the cell wall looks like. You have your bacterial cell. It has a membrane that separates it from its environment, but then it also has the cell wall that separates it from its environment as well and protects it from its environment. That cell wall is made up of these nitrogen sugars. And so remember when we discussed NAG and NAM? And so you have these alternating light and dark kind of turquoise beads. And then those are all connected by peptide linkages. And so these are just really short kind of peptides or pieces of proteins that link those different layers together. It's really essential that you have those linkages, or otherwise you'll just disrupt that complete integrity of the cell wall, and you won't be able to maintain that layer with or around the cell. Okay, and so there are a number of different ways that antibiotics can kind of interact with and inhibit the formation of or the integrity of the cell wall. We're going to specifically look at these two antibiotics and what they do. Okay, so here we go. The first is the beta-lactams. Specifically, the example that we're using is penicillin, but all of the beta-lactams have this beta-lactam ring that you can see here and here and here and here. And what that ring does, it is actually interacts with the enzyme that forms those linkages between the various layers within the peptidoglycan cell wall. And so here it's a transpeptidase. It would normally, naturally, it would connect the two parts of that short peptide, and then that would allow for the cell wall to kind of all be connected. Now, see, this is what the normal kind of connections would look like. But any beta-lactam, including penicillin, when it comes in, it instead binds to the enzyme. And that's a non-reversible binding to that enzyme, and then the enzyme is no longer able to do its function, and then those peptides never get connected, and as the cell grows and grows or attempts to reproduce, then those linkages are not formed, and the complete integrity of the cell wall is damaged, right? And essentially, eventually, you start to lose cell contents because that cell wall is no longer protecting the cell and no longer maintaining the integrity of the cell, and the cell dies. Okay. Now, vancomycin, which is our lipoglycopeptide, it does something similar but different. So again, we have these peptide linkages that need to be connected to have that integrity, right? What vancomycin does is it's a large antibiotic that actually just sits. It literally binds to those peptide chains. And then when this enzyme comes by to try to link them together, they're not available for binding. They're not exposed for binding because that vancomycin is literally holding on to them. And then the impact is the same. Let me move that back. The impact is the same in that you cannot create those cross bridges between the cell walls, the layers of the cell wall, and then you do not have that complete integrity within the cell wall. And you essentially have this happen, cell lysis. Okay, so in both examples, those cross bridges, their integrity is compromised, and then eventually as the cell grows, or tries to reproduce, then it will die. Now, both of these are dependent on the cells being actively growing and reproducing. Static cells that are in the static phase of their life cycle, will not actually be impacted as much by either penicillin or vancomycin because they both, like we said, they kind of prevent the formation of future cross bridges, but they don't destroy old cross bridges, right? And so in that case, both of these are actually dependent on the cells growing and reproducing. Okay. Which is what we just mentioned. And there are other antibiotics as well that do similar functions, but in eukaryotic cells like fungi. So a quick note on antibiotics that inhibit the cell wall formation. Beta-lactams tend to be more broad spectrum, and they have coverage of both gram-positives and gram-negatives, although some gram-positives have actually lost their responsiveness to beta-lactams like penicillin. And that's essentially through developing resistance to those beta-lactams. And actually what's happened is bacteria produce enzymes that can then, it's called beta-lactamase. It's an enzyme that will actually break down or degrade any beta-lactam in its environment, okay? And so for those examples, for those mostly gram-positive organisms, that's why we use vancomycin instead. And so vancomycin is still very effective against most gram-positives, and specifically MRSA, right? Which is your methicillin-resistant staph infections. Now, the next mechanism that we want to look at is the protein formation within cells, okay? And so we've looked at, antibiotics that destabilize the cell wall. Let's talk about some of these mechanisms or metabolic processes within the cell and how they're interrupted by antibiotics. Okay. Just a note here, I'm actually following the same flow as the chapter in your book in the various kind of mechanisms in order, so you should be able to follow them, even though the slides are a little bit different from the original slide deck. Okay, and so this next mechanism looks at antibiotics that inhibit protein formation. So just as a review, if we remember, we have ribosomes, which have two parts, a larger subunit and a smaller subunit, and those essentially cradle this mRNA molecule that then goes through and is read off, and you have your tRNAs that come in and bring an amino acid, and then that amino acid is added to the chain, so on and so forth, until you get to a stop codon, and then those two subunits desegregate and move on to the next mRNA, molecule. Now, this is actually looking at antibiotic impact of antibiotics, but that just gives you an idea, okay? And so for the antibiotics that we're talking about, they specifically impact the smaller ribosomes that are within prokaryotes. So the 30S subunit or the 50S subunit of prokaryotes, which is how they're selectively different from the host, which has these larger ribosomes, which will, generally speaking, be unimpacted. There are some antibiotics that actually do have an impact on the human host because the antibiotic is actually toxic to the liver as well. And so generally speaking, these specific types of antibiotics will be basically used carefully. Okay, so let's look at these. So there's three categories of antibiotics that work specifically on protein formation that we'll talk about: macrolides, aminoglycosides, and tetracycline. And the example of aminoglycosides is streptomycin. So your macrolides, which are these guys here, they actually bind to the larger subunit, the 50S subunit of the ribosome, and they inhibit, essentially, they stop the mRNA from being able to move forward, and that stops protein synthesis, okay? So those are our macrolides. Macrolides tend to be mostly effective on gram-positives, okay? And that's what they're used for, infections of gram-positive bacteria. Oh, let's get that back in order. Then you have aminoglycosides like streptomycin, and these aminoglycosides, as well as tetracyclines, they actually impact the smaller subunit of the ribosome. And for your aminoglycosides, what happens is it binds to the 30S subunit and actually changes its shape just a little bit, so it impairs the reading of the codons, and it essentially just messes with the protein formation thereafter. Whereas the tetracycline, it also binds to the 30S subunit. Instead of just changing its shape, it actually stops the mRNA from passing through. So it kind of has a similar mechanism from macrolides in that it stops protein codon reading, and in that way it stops protein synthesis. And so you completely inhibit protein synthesis in that way. Aminoglycosides tend to be used for gram-negative bacterial infections, just, generally gram-negative bacterial infections. And tetracycline tends to be used for both. It's a more broad spectrum antibiotic that's used for both gram-negative and gram-positive antibiotics. Okay. So that's protein synthesis and our three examples: macrolides, aminoglycosides, and tetracycline. Okay. So let's go ahead and talk about antibiotics that actually impact the cell membrane. Okay. So if we remember, the cell membrane is under the cell wall, and it is essentially, it's very active, right? It's not just like our cell wall that it's a protective layer. It's a very active phospholipid bilayer that has many different proteins and various types of molecules within it that all help to maintain the influx and outflux of various chemicals to and from the cell. And it's a very active membrane. And that membrane, it maintains the integrity of the cell, and it maintains the metabolic processes within the cell. So anything that perturbs that membrane is going to lead to eventually cell death. And that's exactly what these antibiotics do. So we're going to talk about polymyxins in this example. And polymyxins, essentially, they almost kind of create a pore. That's literally what it is. They create kind of a pore through the cell membrane, and a number of those in the cell membrane, and then that membrane just loses its integrity and that cell dies. Okay. One note on polymyxins is that it's also a chemical that can impact the kidneys of the host. And so it's useful against gram-negative infections like Pseudomonas infections, but it should be used very carefully because it also, in the long term, can have impacts on the kidneys of the host. Okay, and so let's look at how this works. So this diagram here is specifically for an antifungal drug, but polymyxin works in exactly the same way, in that you have this long chain that mimics some of the chains in the phospholipid bilayer, but instead of maintaining that integrity, they actually puncture these pores through the cell membrane, as we had just said, and those pores eventually allow for the environment to kind of influx into that cell, and then that cell lyses and the cell dies. Okay. So that's polymyxin, and we said that polymyxin is effective against gram-negative bacteria, specifically Pseudomonas, but it also can have impacts on the kidneys as well. And we're going to talk more about some of the side effects and impacts of antibiotics in our later discussion in this chapter. So the next mechanism for action that's mentioned in your book is to look at metabolic activity. And so you do have a number of antibiotics that can focus in on inhibiting various metabolic processes within prokaryotic cells. And once you inhibit those, then that can actually lead to either the cell not being able to function correctly or eventually dying. We're going to specifically focus on sulfonamides, which are kind of inhibit the folic acid synthesis process, but there are other ways to inhibit metabolism as well. For example, inhibiting electron transport chains, or deactivating enzymes, or destabilizing various proteins that are part of the cell, the cytoskeleton of the cell. So there are many ways that you can do this. We're also going to give an antiviral example here, but we're going to focus on sulfonamides. And so essentially the reason why sulfonamides are so useful is that humans, we have to intake folic acid in our diet. whereas bacterial cells and prokaryotic cells are actually able to produce their own folic acid. So we don't have a folic acid pathway, but bacteria do. And so if you inhibit the folic acid pathway, then you're less likely, or the impacts of the antibiotics are less so on the host. And so what is this folic acid pathway? Essentially, folic acid is important for many different kind of metabolic processes within the cell, including the production of genetic information. And so what you start off with is you have this molecule, which is our PABA molecule, or para-amino-benzoic acid. And essentially what that molecule does is it's a precursor for folic acid that goes through a metabolic pathway with many different enzymes. And depending on where it's going to be kind of active within the metabolism of the bacteria, it could be processed more kind of, throughout this pathway. And so what sulfonamides do is there are a number of sulfonamides, and you don't need to know the different sulfonamides, just know the category. And they're analogs of this PABA molecule. So here, this is the PABA molecule, and these are the sulfonamides. And you can see the shaded area has very similar structure between all of these different molecules. And so what are they doing? These antibiotics are pretending to be this precursor molecule. And so once they're released into the body and then released into the cell, this is what happens, is that all of these sulfonamides, or whatever sulfonamide it is that's being taken as the antibiotic, then interacts with the enzymes that are part of this folic acid synthesis pathway, and they just block the enzymes, because they're not actually the precursor for folic acid, and then that shuts down that pathway, and eventually the cells will no longer be able to grow and reproduce. Okay, and so those are our sulfonamides. We also have this example of protease inhibitors. And so these specifically inhibit enzymes that are important to HIV to be able to replicate within a cell. And so they kind of do the same thing where they come in, they attach to those enzymes and they inhibit them, and that stops the replication cycle of HIV and can kind of modulate some of those symptoms within HIV patients. So our next mechanism for action for antibiotics is the inhibition of nucleic acid synthesis. Now, in your diagram, you have DNA replication and RNA synthesis. We're actually going to combine those together here because there's some overlap between the two of them. Okay, and we're going to be looking at our rifampin and our quinolones, both in this category. So we all know the essential nature of genetic information, right? We talked about the central dogma of biology. If you don't have genetic information and the ability to transcribe and translate and replicate that genetic information, then you're not going to have metabolic processes within an individual cell, and you're also not going to be able to reproduce and continue those populations into future generations. So really, any chemical that inhibits nucleic acid synthesis overall, generally, whether it's the replication of DNA, the transcription of RNA, or the translation of that RNA into proteins, which we've already talked about, then that's going to impact the cell in terms of its metabolic processes and its growth and reproduction. And so the two chemicals that we'll focus on are quinolones and rifampicins. And so what you're looking at here is actually transcription. And so you can see that here we have the DNA molecule here, and then the copy of RNA is being made here. And you have your RNA polymerase here, and then you have your gyrase molecule here. If we remember from transcription, our discussion of replication and transcription, both processes in bacteria need DNA gyrase. And what that is whenever you're unwinding the DNA so that you can make copies of it or you can replicate it, it's really essential that you don't have supercoiling, right? So if you remember like old-time telephones, if you were to kind of knot that spiral cord together, then it would supercoil or get knotted on each other. And so as you unwind the DNA, that can happen as well. And so the DNA gyrase goes in and stabilizes those coils so that they don't supercoil. And so our first amino acid, quinolones, they actually inhibit DNA gyrase. And so that just makes a mess of the supercoiling of the DNA. And eventually that DNA is not accessible, and then that cell is not able to maintain its functions. Whereas rifampicin is specifically interacting with the RNA polymerase, and that's actually impacting the transcription. Okay, so your quinolones are impacting both replication and transcription, whereas your rifampicin is just impacting transcription. Quinolones tend to be useful against both gram-positives and gram-negatives, whereas rifampin is actually pretty specific. It's pretty specifically used against tuberculosis infections, and then meningitis that's specifically caused by various species of Neisseria. And so it's a pretty kind of specific, it's not a broadband antibiotic. Okay. There are other ways that you can impact nucleic acid synthesis, replication, transcription, and other mechanisms as well by just jumping into the process at the various kind of enzymes along the pathway. Okay, but that's what we'll focus on here. And we're going to jump back into the last mechanism that's on our diagram, which is attachment and entry. Attachment and entry is actually mostly related to viruses. So this discussion here, we're going to look at a couple of different antiviral drugs and how they impact viral attachment, okay? Or viral attachment and viral kind of processes. And so just a reminder, you have viruses, they attach to some sort of receptor on the host cell, and then they're incorporated into the cell. Once they're incorporated, their genetic information has to be released. And then that genetic information can then be utilized to make more viruses. Now, their antiviral drugs will inhibit various different steps along this process. Attachment, uncoding, which is right here. You can inhibit uncoding, which is our next example. You can inhibit the actual production of proteins, and you can inhibit the release of the virus. And so we're going to focus just on antiviral. This antiviral specifically inhibits the binding of various types of cold viruses to the receptors that allow them to enter into the cell. It just binds onto those receptors, and when it's bound, then those cold viruses are not able to actually enter the cell, and then you can prevent infection. So our next example is a Rildrun, and what this is doing is actually allowing the virus into the cell. This is specific to poliovirus. The virus is allowed into the cell, but once it gets into the cell, the proteins that are needed for uncoding that virus and releasing that genetic information into the cell so it can actually go into its lytic cycle and become virulent are inhibited, and therefore that virus is not allowed to become virulent, and it's just kind of cleaned out of the cell. And so these are kind of all of our different mechanisms for inhibiting microbial pathogens once they've actually kind of infected its host. We talked about inhibition of the cell wall. We talked about inhibition of protein synthesis. We talked about disruption of the integrity of the cell membrane. We talked about inhibition of metabolic processes, specifically focusing on sulfonamides. We talked about inhibition of both DNA replication as well as transcription. And then we talked a little bit specifically about viruses. Okay. And so this kind of gives us an overview of how different types of clinical drugs work. So when you're choosing a clinical drug, what are the considerations that you're going to take to kind of define which is the drug to use? Now, there's a lot of considerations. We won't talk about all of them, but we will kind of go through some general ideas. If at this point you want to pause the video, go back and review the different mechanisms and the examples that we use, whether they work on gram-positives, gram-negatives, which types of specific organisms they work on, which we will actually get back to in a little bit in a couple of slides, then go ahead and do that. Okay, welcome back. So we already said that it's very important that antibiotics be selectively toxic, so that they are toxic to the pathogen and not to the host, preferably that they are toxic to the pathogen and not to the microbiome of the host, although that is more difficult. Also, it should be readily available and easy to produce, inexpensive so that it's accessible, and it should be chemically stable, which means that once you produce that antibiotic, it should have a shelf life. It shouldn't actually start to, change over its shelf life. Should be easily administered and kind of mechanisms that we're familiar with, tablets or IVs, so on and so forth, and it should not produce extreme or side effects. Okay. So these antibiotics, some of them have a narrow spectrum, some of them have a broad spectrum. And you're already familiar with that because as we were going through them, we said this antibiotic is effective against both gram-positives and gram-negatives, for example, or this antibiotic is only effective against, tuberculosis, or this antibiotic is only effective against gram-negatives. And so that gives you an idea that some of them function across many different species that cause infection, and some of them are more specific. And so what we had looked at in our discussion are broad spectrum antibiotics that are generally effective against both gram-positives and gram-negatives, including penicillin, tetracycline, various types of sulfonamides, and quinolones. Okay? Then you have antibiotics that are specific just to gram-positives, and those are your lipoglycopeptides, like vancomycin, and that was actually developed specifically for gram-positives, specifically for MRSA or Staph aureus strains that are actually resistant to other types of antibiotics. And that would be your vancomycin, so specifically for gram-positives. Also, macrolides are specifically for gram-positives. Then you have antibiotics that are just more specifically for gram-negatives, like your aminoglycosides and your polymyxins. And then you have even really narrow spectrum antibiotics, like rifampin, which is specifically for tuberculosis, and some infections of meningitis that are specifically caused by various species of Neisseria. Or your antivirals that are specific to the virus that they work against. And so you can see how we go from really broad spectrum to really narrow spectrum. Another thing is that oftentimes antibiotics are given within combinations or cocktails so that you could try to get at various aspects of those kind of impacts that we had talked about, impacting nucleic acid, a formation and protein formation and the cell wall or the cell membrane, so on and so forth. So you can try to get at all of the different pathogens and all of the different strains and not leave behind antibiotic resistant strains. So how do we know? How do we know if something is effective? How do we know if something is targeting the specific organism that we want to look at? We have a number of various tests. And actually in our microbial control in the environment chapter, we talked about some tests to look at chemicals and how effective they are against various types of microorganisms that we're trying to control in the environment. There are some similar tests that we can use. So one test is this minimum inhibitory concentration test, which is very similar to what we talked about in chapter 9, where you make different concentrations of an antibiotic and you expose bacteria to those, and you literally just look for growth. So anytime you see kind of a opaque cell, then that means there are bacteria that are growing in that cell. And then these cells that are clear, there's no bacterial growth, okay? And so I would assume that these are positive controls here, negative controls here, and then serial dilutions of various types of antibiotics here that have been inoculated with different types of the organism that's being targeted. Not different types of organisms, but the organism being targeted. And wherever you see a concentration where there's no growth, then you know that that's kind of the concentration that you want to use, or that would be effective in this case. Okay? That's once you actually know which antibiotics that you want to use. There's an initial step, though, to see which antibiotics are actually going to be effective against that specific organism that you're trying to kind of control. So oftentimes, you'll get a culture off of a sick individual, and you'll isolate the organism, that's the pathogen, and then you'll actually expose it to various types of antibiotics. You will do this in your microbiology lab, and you'll see which has the largest zone of inhibition. And this is just the halo that's around that antibiotic that indicates cell death in that area. Now, those antibiotics that are effective, then you will look at some of these other characteristics that we'll talk about in a minute. What's the level of effectiveness of the antibiotic? So this minimum inhibitory concentration test, what are the side effects, so on and so forth. You're not going to be able just to choose the antibiotic with the biggest zone of inhibition, which is the most effective, because you also have to consider other characteristics of that drug. Does it have side effects? Is this a child? Is this a pregnant woman? So on and so forth. And so you then make that distinction. Okay. You can also look at how that antibiotic is administered, topical, oral, intravenous, for example, and look at side effects of the various drugs. So these are antibiotics. They are against life, right? That's their goal, is to inhibit the growth of a living organism. And so sometimes they do have side effects. We already talked about a number of antibiotics that impact the liver and kidney. Generally speaking, antibiotics can have a higher impact on children and pregnant women, or the fetus specifically. And so what we do is we'll actually look at the effectiveness of the antibiotic and also the toxic effect. And then this therapeutic index is generated that allows you to indicate which is the most appropriate antibiotic for the specific infection that you're looking at. Okay? Because you want to prevent side effects. You want to prevent if somebody is allergic to a drug, so on and so forth. Okay? So antibiotic use is essential because it allows us to control microorganisms that are pathogenic in our bodies. But we already know, because we had some discussions around antibiotic resistance previously and also in this lecture as well, and we're going to continue those discussions now. Antibiotics are essential tools, but because of the processes of evolution, microorganisms and pathogenic agents are able to kind of modulate their characteristics based on what we throw at them. And so we know this because we're in a pandemic and there are numbers of booster shots and various waves of the infections that we see. But essentially, antibiotic resistance is when an organism is able to either pick up a gene from their environment, right, through horizontal gene transfer, or there's a mutation that it has in its own genetic information that allows it to become resistant to one of these antibiotics. Okay. There's different mechanisms for developing antibiotic resistance, and there's kind of different ways that antibiotic resistance can be had. And so let's look at those.
A big challenge to medical science today is the growing problem of antibiotic resistance. A pathogen that is resistant to a particular antibiotic will not be killed or inhibited by that antibiotic. It has gained that resistance either by random mutation or through gene transfer from another bacterium. In a particular population of bacteria, there may be a few cells that are resistant to the effects of a particular antibiotic. When the patient is given a particular antibiotic, that drug will kill or inhibit the growth of all the sensitive bacterial cells. This allows the resistant cells to multiply freely in this less crowded environment. One way that bacteria can acquire resistance genes is through mutation. If a mutation occurs in a sequence of DNA, this could result in a different amino acid sequence. Consequently, this different sequence could produce a new enzyme, perhaps one that can interfere with the action of an antibiotic. Bacteria can also acquire resistance genes by horizontal gene transfer. One type of horizontal transfer is transformation, in which a cell picks up a piece of DNA from its surroundings. Or a bacteriophage can transfer the resistance genes from one bacterial cell to another in the process of transduction. Finally, resistance genes can often be found on small pieces of DNA called R plasmids or R factors. These R factors are often transferred via conjugation between bacterial cells. Bacteria can resist the effects of antibiotics through several mechanisms. They may secrete enzymes that destroy the chemical structure of the antibiotic. For example, penicillin-resistant bacteria produce an enzyme called beta-lactamase. The beta-lactamase enzyme breaks the beta-lactam ring in the molecule of penicillin and related antibiotics, such as cephalosporins. This prevents the antibiotic from disrupting the peptidoglycan cell wall structure. Other bacteria prevent antibiotics such as tetracycline from passing through their cell membranes. They do this by changing the structure or charge of pore proteins. Even if the antibiotic can penetrate the cell, a resistant bacterium can make it impossible or more difficult for the antibiotic to bind to its target. Sulfonamides are antibiotics that compete successfully with PABA for an enzyme binding site in the folic acid synthesis pathway. Resistant bacteria may have evolved an enzyme with greater affinity for PABA, thus making the sulfonamide less effective at blocking the folic acid pathway. Other resistant bacteria have developed a method to obtain molecules they normally synthesize directly from their environment. Therefore, they aren't affected by the blockage of the biosynthesis pathway. Finally, some resistant bacteria have membrane-localized efflux pumps that remove some antibiotics from the cell. Some resistant bacteria have a greater number of efflux pumps, thus increasing their ability to eliminate the antibiotic. In some cases, bacteria have also been able to increase the spectrum of drugs that are eliminated by these pumps by making pumps less specific.
And so those two discussions give you an idea of the issue with antibiotic resistance and some of the mechanisms for antibiotic resistance as well. And so when it comes to antibiotic resistance, the CDC has 3 levels of urgency. Urgent, which is widespread and life-threatening and oftentimes multi-drug resistant. So organisms that are strains of organisms that are resistant to many different drugs. Right now, Clostridium difficile is on this list of urgent, potentially life-threatening diseases. Serious the serious kind of level is also makes it's a pathogenic organism that leads to disease and is widespread. And MRSA or Staph aureus is on this list, specifically various types of antibiotic resistant Staph aureus. And then there's concerning where you do have a pathogenic organism, but its impact or its how it's spread through a population or through the world is less so. And so you have different types of antibiotic resistant groups of Streptococcus that fall under this grade as well. Okay, we just mentioned multiple drug resistance here, and we had mentioned in that video the beta-lactamase gene that basically inactivates penicillin and beta-lactams. Okay, so what to do? And that's how I want to end this lecture here, is what to do. So we know the processes that allow for us essentially to develop antibiotic resistance. You have these organisms, you try to control them in the environment, one, and then some of them are able to resist that control, which we had talked about in chapter 9. And then somehow they're able to actually infect organisms, which is one of the discussions that we're having as well. And then you have pathogenicity in those organisms, and then you again expose them to antimicrobial agents, antibiotics, and those kill many of those organisms, but leave the most resistant ones to have the opportunity to continue to grow and reproduce. And so the use of antimicrobial agents in the environment and antimicrobial drugs is really driving in a lot of ways the evolution of antimicrobial resistant microorganisms. And so considering these factors, right, using antibiotics carefully, right, being thoughtful of when they're used. For example, many antibiotics have been prescribed for the common cold. The common cold is a virus, right? So that is antibiotics that have been used, essentially suppressing many microorganisms in a person's body, but allowing for the most resistant to continue to grow and reproduce. And then that over time leads to the production of antibiotic resistant organisms when you didn't even need to use that antibiotic in the first place. Okay, so really using antibiotics sparsely. The same goes for microbial control in the environment, being thoughtful of how we use various types of disinfectants and things like that in an environment so that we're not encouraging the growth of antibiotic resistant organisms. When it says it kills 99.9% of microorganisms, that 0.1% is the one that goes on to evolve and mutate and generate antibiotic resistance. Then, in cases that are determined that antibiotics are important and they should be used, making sure to use the full drug over the full period that is prescribed by the doctor. Because what's happening is you're exposing the infection to that drug over time. And over time, the more and more resistant organisms are the ones that are maintained in the body. And so if that drug, somebody stops it because they're feeling better, and they're not actually using that drug to completion, then they're actually cultivating those more resistant organisms in their body, right? Then it is very common to use antimicrobial agents in combination where you have multiple drugs that are used together to try to, get at various aspects of that organism's growth and metabolism to kill as many off as possible and not leave resistant strains within the body. Then there is the development of new antibiotic drugs, which is really essential. It's absolutely essential. But if we don't do these previous steps, then it is kind of a rat race because these organisms will continue to mutate and continue to develop resistance to the new drugs as we develop them. And so we would like to have, as humans, have the advantage by utilizing these drugs effectively so we have more useful antibiotics that are not, organisms have not become resistant to, instead of having to just continue to produce more and more semi-synthetic or synthetic antibiotics over time. And so this is a book kind of continues this discussion as well and gives some details, so make sure to go into that. But this is kind of the general guidelines for how we can try to be thoughtful and effective in our use of antibiotics so that we're not actually leading to the development of antibiotic-resistant organisms or microbial agents within the environment, within our bodies. So that is the close of chapter 10. You should have been taking notes throughout the discussion, maybe an outline, maybe a flow chart, maybe a diagram. Just take an image of parts of that note. You don't necessarily have to share all of your notes with the class, but show that you've created some sort of flow chart or diagram, some sort of outline to help you kind of keep all of these different concepts in line, and put that into the second link in the description for points. I just want to make sure that it is indeed the second link. Yes, the second link in the description for points. And I'll see you in the next video.